It is now my pleasure to introduce Dr. Wenlei Jiang, Senior Advisor for Innovation and Strategic Outreach in the Office of Research and Standards at the Office of Generic Drugs. Dr. Jiang is a recognized leader in complex product research, global harmonization of bioequivalence standards, and scientific outreach. She also plays key roles in international regulatory initiatives, including serving as a U.S. co-chair for Global Bioequivalence Harmonization Initiative. Today, she will be introducing Session 1, Insights into Product-Specific Guidance for Complex Products, from Research to Standards. Please join me in welcoming Dr. Jiang.
Yeah, thank you, Maria, for your kind introduction. Good morning, good afternoon, good evening, everyone. Welcome to Session 1 of the 2025 Advancing Generic Drug Development Workshop. I'm Wenlei Jiang from Office of Research and Standards, Office of Generic Drugs, CDER, US FDA. Dr. Nahid Kamal from Office of Pharmaceutical Quality and I co-led the planning of this session. It is my great pleasure to introduce today's first session titled Insights to Product-Specific Guidance for Complex Products, from Research to Standards. In this session, we will explore how cutting-edge FDA intramural and extramural research translate into practical science-based guidance that supports industry in developing safe, effective, and high-quality complex generic products, a comprehensive journey from research to standards. First, we will briefly review FDA's product-specific guidance program and its critical role in supporting complex product development, showcase the innovative subject matter expert triage team, abbreviated as STT, that proactively identifies research needs and streamlines guidance development for complex products. We will also present real-world case studies demonstrating how research directly informs regulatory standards and highlight efficient bioequivalence approaches that reduce regulatory burden while maintaining scientific rigor. As of October 2025, FDA has posted over 2,300 product-specific guidances. In this session, you'll discover how FDA has transformed its approach to meet GDUFA 3 commitments. Particularly noteworthy is our progress with complex products. The cross-office STT CDER established since the beginning of GDUFA 3 has made complex PSC development timeline more transparent and predictable. We will explore fascinating case studies spanning different therapeutic areas. For example, roflumilast, a topical cream, demonstrating how research resolved critical knowledge gaps, complex collagen implants, showcasing Excipient Sameness challenges. In addition, for locally acting dry powder inhaler and meter dose inhalers, FDA has increased the percentage of the PSG offering alternative approaches to clinical endpoint studies significantly. For topical products applied to the skin and mucosa, there is also significant increase in the number of PSG providing characterization-based BE approach. You will hear these specific updates from our speakers soon. So leading today's session are 4 distinguished speakers who represent the breadth of FDA's expertise. First, Rangita Kumari, a project manager in the Office of Pharmaceutical Quality Research, oversees strategic coordination of our STT initiatives, ensuring efficient knowledge management across CDER offices. Dr. Nahid Kamal, our research pharmacologist from the Office of Pharmaceutical Quality, leads cutting-edge research and complex drug delivery systems, including transdermal topical, and implant products. Her work directly bridges laboratory science with regulatory guidance development. Dr. Liang Fenghan, a clinical analyst from Office of Research and Standards, Office of Generic Drugs, brings extensive expertise in bioequivalence study design and clinical efficacy assessments, particularly for orally inhaled drug products. Dr. Megan Kelton, also from ORS OGD, a senior pharmacologist specializing in topical and transdermal drug delivery, has been instrumental in developing efficient bioequivalence approaches that significantly reduce regulatory burden for multiple strengths products. So today, in the first session, from implant products and inhalation therapies to topical formulations, Session 1 presentations will demonstrate how FDA's collaborative, science-driven approach is guiding complex generic drug development more efficient. You'll see firsthand how our research investments translate into practical solutions that benefit both industry and patients. Without further ado, please join me in welcoming our first speaker, Rangita Kumari. Rangita, the floor is yours.
Thank you, Wenli. Good morning, everyone. I'm Rangita Kumari, and I'm the project manager in Office of Pharmaceutical Quality Research. Today, I will provide an overview of product-specific guidance program and focus on translation of research findings to support the development of product-specific guidance. As the title states, I will limit the scope of my presentation on complex product as those are more challenging. In my next few slides, I will cover product-specific guidance program for complex products, as well as the SME Triage team program the approach that FDA has taken to proactively identify research need for complex product. I will also share an example of a case study, Roflumilast topical cream, to illustrate the process. So let's talk about the product-specific guidance program. As you may know, this product-specific guidance PSG started in 2007, aims at facilitating the development and approval of safe, effective, and high-quality generic drug product by providing FDA current thinking and recommended approach for demonstrating therapeutic equivalence of a generic drug to its reference-listed drug product. PSCs are posted on quarterly basis on FDA website. And as you can see here, as of September 2025, there are over 2,200 PSC already posted. It's important to note here that PSC are FDA current thinking. That means ANDA applicants can provide alternative approach that are deviating from the recommendation in product-specific guidance, but they need to also provide the justification for the proposed approach, as well as including the data or any references. PSC enhances the transparency and expectation between the industry and the generic industry. It also reduces industry inquiries by providing a general framework for the drug development. Last but not the least, it also improves the quality of submitted ANDAs. As you can see here, there are challenges associated with the development of product-specific guidance, especially for the complex products. Excuse me. So there are about 100 of new NDA approval, complex NDA approval per year that warrant the development of product-specific guidance. Out of those new approvals, approximately 30% of the new approvals are considered to be complex drug product. And these products may include different route of administration and can meet more than one definition of complexity. The complexity could be due to complex active ingredient, complex drug device combination, or complex dosage form. To help industry with the development of PSG for complex drug product. As negotiated in GDUFA 3, FDA has agreed to provide on priorities and goal dates for the development of this PSG. As you can see here, per the commitment, GDUFA 3 commitment, a PSC will be posted for the 50% of the complex NDA within 2 years after the approval date, and for the 75% of the complex NDA within 3 years after the approval date. As you can see here, those are very aspirational goals. To meet this requirement and meet this commitment, FDA needed to come up with a very efficient process that can identify any knowledge gap and any early in the process. This is where the SME triage team process comes into play. A proactive approach that helps to identify research need right after the approval, particularly for the complex products. Prior to the rollout of GDUFA 3, FDA established a process called SME Triage Team Program to support the development of a PSC for complex product. Please note that this process is particularly focused for complex product. This clarifies for this process, we have the SMEs, the relevant subject matter expert from different offices, including both Office of Generic Drug and the Office of Pharmaceutical Quality. The one key benefit of the process is that this connects the researchers directly with the assessor and as well as the policy staff, and gives an opportunity to discuss about the complexity and any knowledge gap associated with the product. The areas are designed and created based on the SME teams are created, subject matter teams are created based on the area of complexity, and those come from across 9 different CDER offices, and the staffs are from research, policy, and the review. The SME team members are also tasked with making key decisions in terms of discuss the complexity of the product, identify any knowledge gap, and also if the knowledge gap is identified, the team collectively decide what should be the objective of the research to generate the evidence that can be put into the recommendation. So as you can see here, this cross-disciplinary process enables the effective knowledge management and maximizes center resources. This process we piloted in 2021 and became fully operational after the implementation of GDUFA 3, October 2022. Some high-level overview about this program. As I mentioned, this is an internal program to identify and direct research to support PSC development for complex generic drug product. The program consists of 10 complex areas based on the area of complexity, and those are subject matter comes across 9 CDER offices. As you can see here, the number states here. So far, since the beginning of the process, we have triaged 96 complex NDA through this process. The team has already identified there are 15 complex NDA that need further research to generate the evidence to provide the recommendation into the PSG. And out of those 96 NDA, for 65 of those NDA, the PSG has already been published. This is a high-level overview of the process in terms of the timelines. So as you know, this complex product, from the start of the NDA approval, it is very important for FDA to identify the complex NDA, because as we know, the research could take very long times. Right after the identification of the complexity, that's where the SME subject matter expert team sits together and decides if there is a need is there any knowledge gap, and discuss the challenges associated with the process, and then narrow it down to the products that need further research to generate the evidence. After identification of the NDAs for the research, we conduct the research in our lab, or it could be a research project within here in FDA in our OPQR lab, or it could be awarded external. After the completion of the research, we translate the research findings into the recommendation, and that recommendation finally gets put into the guidance for the industry for their reference. So since the data we have so far, looking at the data from last 5 years, about 26% of the new approvals are complex in nature. And as you can see here, approximately there are 5 to 7 new complex NDA approval per quarter, as you can see from the bar chart below. Out of all those complex NDA, there are about 16% of those complex NDA further need research, meaning there are some gap in a knowledge gap we have seen that needs for research to generate the evidence. And for most of them, as you can see here, those are from the complex API area, for example, oligonucleotide, peptides. They're from complex route of administration, topical inhalation, as you can see here in the chart. So now let's walk through roflumilast topical cream, a case study to illustrate the process. So this roflumilast topical cream was approved in July of 2022. Right after the determination of the complexity, we gathered the SME, it was assigned to the SME Triage team for this area. And then the team collectively looked at this product from all perspectives, meaning the challenges associated with the product, any complexities, and if there is any knowledge gap to proceed with the PSG development. Based on the discussion from the SME team, the team collectively agreed that it seemed at the time when they were discussing back in 2022 that there were some concerns the team had in terms of two major areas with clinical endpoint-based bioequivalence approach and with in vitro characterization-based PE approach. So it was decided that we need to do further research to generate the evidence and answer those questions before FDA can provide the recommendation into the guidance. So the research was conducted in OPQR, Office of Pharmaceutical Quality Lab here in FDA. The main purpose, the goal, objective of the research was to investigate the permeability testing of roflumilast across human cadaver skin and to assess if IBPT can be a component of characterization-based in vitro B approach. And then the second objective was to identify and characterize the observed crystals, analyze the root cause of the crystal formation, and assess the drug product's thermodynamic stability. After completing the research was successfully addressed the concerns that the SME team had at the beginning of the process, which helped actually support the development of the PSG and also helped with the regulatory decision-making by resolving key scientific uncertainties through comprehensive FDA research, establishing validated IBPT methodology, and correcting crystal identification misconception. The research that we conducted in lab actually also helped enabled with the efficient regulatory actions by providing essential scientific foundation from research findings for PSG drafting and also supported the multiple review of multiple pre-ANDA applications. What I'm trying to illustrate here is, as you can see, the process from the time of approval of this topical cream, which was approved in July of 2022, till the guidance development. Right early in the process, right after the approval, once we identified the complexity, we held the triage meeting in December. Within 5 months of the approval, we held the triage meeting, and that gave team this opportunity to discuss about the product and identify if there is any knowledge gap associated with that product. After the identification of research need, we conducted the research in our lab here within FDA. The research lasted almost about 2-year period of time frame. And once the research was completed, the information that the research finding that we obtained, we translated that research finding into the recommendation, and we put that recommendation into the PSG. And the PSG for this cream actually is already published back in May of this year. The PSG has already been published. So as you can see here from the start of approval, within 3-year period of time frame, FDA has been able to publish the guidance for this complex product within 3 year of time period. So as you can see here, this SME triage team process is very instrumental in identifying the need for, if there is any complexity associated with the process, and identify the research knowledge gap and research need early in the process, and then conduct the research right after the approval. So it helps FDA to meet its commitment for the complex product, as I mentioned, according to the commitment letter 3. In summary, FDA Product Specific Guidance Program provides FDA current thinking on the type of studies and information to support the development of safe, effective, and high-quality generic product. The SME triage team process helps to identify and address any potential challenges or knowledge gap and research need proactively early into the process for the PSG development of complex product. This also helps with this life cycle approach helps with knowledge generation, and information sharing is critical. It's very critical for the timely generation development of PSG, particularly for the complex product. Thank you. Now I would like to introduce our next speaker, Dr. Nahid.
Thank you, Rangita, for the wonderful presentation. I'm going to present another case study that was our collaborative effort of our SME Triage team, mainly focusing on the complex implant products, Excipient Sameness, Characterization, and Bioequivalence challenges. I'm Nahid Kamal. I'm a pharmacologist from Office of Pharmaceutical Quality Research under OPQ CDER FDA. So my presentation will cover 4 key areas. First, I will talk about the RLD, will give an overview on the RLD. Then I will talk about the bioequivalence challenges for the PSG development. And then I will provide our comprehensive research plan that addresses bioequivalence challenges with results and findings. Finally, I will share our current thinking on the PSG recommendation and overall conclusion based on our research findings. So let's start with the RLD, the Xeracol collagen implant, which is approved in 2020 for post-surgical pain relief in adults undergoing the open inguinal hernia repair. For the single-dose administration of this drug product, actually, the drug product comes in 3 implants, where each implant contains 100 milligrams of BBB kinase as locally acting active ingredient in this drug product. The interesting part of this drug product is that it's a mechanism of drug release from the porous lyophilized collagen matrix, which involve weighting, dissolution, and diffusion of the drug for 24 hours at the surgical site directly. So, which demonstrate that the having a good integrity or the adequate architecture of the collagen matrix is very important to have adequate release kinetics as well as the therapeutic performance of the drug product. The implant matrix is composed of purified type 1 collagen, which is the sole inactive ingredient or excipient in this drug product. The complex part of this drug product is that the collagen that used in the RLD is derived from a specific origin or species using RLD applicants' proprietary extraction and purification process. Besides, there is no USP or European monograph for this collagen characterization. If you look at the collagen structure, it has very complex triple helix structure with glycine XY motif, which is glycine, proline, and hydroxyproline. It comes as an ordered sequence. So any change in this sequence, it could be due to the change in species or the change in source of collagen, can significantly impact the property of the collagen, as well as can significantly impact the drug entrapment in the collagen matrix, finally, the drug release kinetics. So keeping those complexity in mind, as Rangita mentioned, whenever any drug approved, so right after that, the PhD Triage team come to a discussion and find out what is the complexities associated with this drug product, which can be hurdle to develop a PhD for this drug product. So what did the team identified that regarding this drug product actually present a significant bioequivalence challenges. For example, for in vivo studies, for conducting a comparative PK study, in the healthy subject, it will be difficult. I mean, it's not feasible because of the invasive nature of the implant administration in the surgical side, so patient enrollment will be difficult since it's a surgical implant, right? And besides, it has a complex excipient. So if you're looking into the drug product which is locally acting, it causes a surgical site dependent PK variability. Now, if you're leaning towards the in vitro B challenges, that poses a equal complexity. For example, there is a knowledge gap regarding the Q1 sameness of the collagen, and we need better understanding how manufacturing process impact product performance, and require a comprehensive characterization method that can be used not only to characterize the collagen but also to understand how the various sources of the collagen impact the product performance. In this scenario, of course, the IV-IVC is not an option for this type of drug product. Now, to address this concern and to fill the knowledge gaps, we have developed a comprehensive three-phase research plan where during the first phase, we have defined the Q1 sameness of the excipients to assess the impact of collagen source on the product performance. Then we have studied, in the second phase, we have studied the dispersions and how manufacturing process like dispersion, mixing speed, mixing time, including the lyophilization parameter can influence the performance of the final product. Finally, we have also studied how critical formulation attributes impacts the drug product, as well as we have developed comprehensive comparative characterization methods to understand the product performance due to change in the collagen sources. So as per the first part of the research, we have characterized the collagen by testing collagen from three sources and named them source A, source B, and source C. These are our test samples. And then we compared the testing result with the reference collagen that extracted from the RLD product, right? Because we don't have access to the collagen that is used in the RLD. And we have used multiple analytical methods to understand the structural, physical, chemical, or thermal properties of the collagen. So it is not possible to go over all the results of the characterizations that we have used for collagens. I will provide a few of them in the next slide. So regarding the structural property characterization results, the structural property of the collagen that we investigated shows a comparable hydroxyproline content, nitrogen over hydroxyproline ratio, free amino nitrogen content in the collagens. And similarly, we found that the FTIR characteristics peak patterns for the collagens behave similarly among this collagen, including RLD, regardless of the variation in the sources. However, differences were observed in terms of the solubility of the collagens, where we found that collagen A shows higher solubility compared to the collagen B, C, and RLD. And regarding the DSC analysis for thermal stability, we found that collagen A shows weaker thermal stability compared to the moderate thermal stability of the collagen B, and collagen C and RLD shows stronger thermal stability. Now, moving to the dispersion characterization as part of the second phase of the study, where we have studied the dispersion as an intermediate product during the manufacturing of the implant. The manufacturing of the implant involves mixing of the collagen dispersion with the API, and then filtration and filling, followed by lyophilization to obtain the lyophilized implant. So during this manufacturing process, every steps involved related to the manufacturing of the implant, we have optimized it during our preliminary studies. So now regarding the dispersion characterization, we have characterized the dispersion to understand its viscosity, structural property in terms of morphology of the fibrillar organization in the collagen dispersion, and the triple helix structure stability of the collagen due to the effect of the manufacturing process. So in this slide, I'm showing the characteristics, dispersion characterization results that was performed by using the morphological directed Raman spectroscopy to observe how fibrillar morphology is in the dispersions due to effect of the manufacturing process. So in the left side of these images, in this slide, you can see the macroscopic image of the A and C, kind of showing similar kind of fibrillar aggregates. The difference is collagen A is kind of little lighter dense, and compared to the collagen C, which is very denser. However, this A and C collagen dispersion fibrillar network is totally different than the collagen B and RLD, which shows similar tangled fibrillar string-like fibrillar network in the collagen dispersion. Then we have investigated the stability of the triple helix structure in the collagen dispersion due to addition of the API or increase in the temperature during the manufacturing. So as you can see in the figure A on the right side figures in the slide, figure A, which is where peak pattern is showing the blank dispersion. Blank dispersion is without API. The peaker actually is showing the characteristics peak, positive and negative peak as 221 and 197 respectively. And when we increase the pH of the temperature of the blank dispersion, the peak pattern did not change that much, which represent that the blank dispersion maintains its native triple helix structure. However, when we added the API into it, the peak pattern significantly shifted, where collagen A and B affected more compared to the C and RLD. And this effect was intensified when temperature was increased from 25 to 37 degrees centigrade temperature, which is showing in the figure D. Then after manufacturing, we have the implant, and this implant we subjected to multiple characterizations method to understand the critical quality attributes of the implants and the impact of various sources of the collagen that used to manufacture this implant. Again, I'm not going to go over all of these characterization results due to the lack of time. So here, the first thing was to characterize the implant formation once we manufacture the implant. As you can see in this slide, the implants are formed completely A, B, C, that manufactured by using collagen from source A, B, C, and compared with the RLD. However, implant A shows a rough surface with noticeable addition of the collagen with the containers compared to the B, C, and RLD, which have smooth surface and some extent of addition to the container. Then we have investigated this collagen implant for morphological, to obtain the morphological information by using scanning electron microscopy. We found that the implant shows distinct pores with irregular organization of the fibrillar structure in the implant, and here in the top, upper tier showing the top surface of the implant, and lower tier showing the cross-section area of the implant. The main part is that this implant doesn't show any similarity compared to the RLD, where RLD shows the honeycomb-like structure. Then we further investigated this implant using micro-CT, which confirms that actually all of these implant A, B, C shows honeycomb-like structure like RLD. Then we further, for in-depth understanding of this collagen implant, the microstructural property as well as the porosity, how it's distributed, we investigated using AI-assisted image analysis after segmentation of the images in collaboration with the DGM. As you can see, the red circles in the bottom figures in this slide, that the red color shows the pores in the implant, and the green color network shows the solid content in the implant, which consists of the API and the collagen. So the result shows that implant A and B has broader porosity and the less solid content compared to the implant C, which has denser network, solid content network, and smaller pores compared to the A and B. However, the RLT shows very dense solid content network with smallest pore and greatest amount of the solid content in the implant. Then we have also investigated how these pores are distributed, the distribution of the pore in terms of watershed color coding analysis. As you can see, the implant A, B, C have different sizes of the pores, but obviously this result, the D10, D50, and D90 pore size distribution is agreeing with the porosity result that I have discussed. Then we have investigated, measured the API distribution in the implant. So we have used the post-AI analysis, image analysis, and segmentation to understand how API is distributed in the implant. In this case, because of the resolution that we have used, it was difficult to differentiate the API from the collagen. However, instead of that, we have taken the collagen, solid collagen content, which consists of the API and the collagen network as an indicator to measure the thickness to understand the API distribution in the implant. So the results show that ABC actually shows the similar value of 4 micrometer thickness of the solid content, solid wall thickness. Here, implant B shows slightly higher than A and C, but these values are statistically non-significant. On the RLD, it shows 6 micrometer of wall thickness. Then we have also used in-house traditional method by using HPLC to understand the distribution of the drug in the whole implant or in the fraction implant. In both cases, the results show that the drug is uniformly distributed regardless of the sources. There is no sign of accumulation in the fraction implant. Then we investigated the solid state API in the implants. The results show that the percent crystallinity in the RLD was significantly higher compared to the C, implant C, and implant B, and implant A in the descending order. And then we investigated the mechanical strength of the implants. The results show that, again, the RLD shows significantly higher rigidity compared to the implant B, implant C, and implant A in descending order. We have also investigated the enzymatic degradation of the implant. The collagenous activity result shows for the implant B was higher compared to the implant A, and implant RLD, and C shows the lowest collagenous activity. And these values are found statistically non-significant. Finally, the drug release study to understand the effect of collagen sources on the performance of the implant. We have conducted the drug release study using ASP apparatus 2 and using a custom-made sinkers. We have investigated different configuration of the sinkers. Here, the results are providing using 10 by 10 custom-made sinkers. And this sinkers was used to hold the implant inside the release media, which was PBS buffer pH 6.8. So that the floating of the implant can be prevented during the release study. So the results show that for all the implant, we have observed a biphasic release pattern where at the initial time point, higher drug was released from the implant A, followed by implant B, implant C, and RLD. At the end of the 6 hours of the release study, the pattern remained same. However, the drug release from the implant B shows higher compared to the implant A, and then implant C and RLD drug release remained same. And at the end of the 6-hour release study, we have examined the residual implant. We found that implant A and implant B actually lost significantly their structure compared to the implant C and RLD, which almost remained intact. So another important information I want to share that our release study that we have performed following the experimental conditions described here was directly positively correlated with the porosity result that I have described in the earlier slide, as well as the negatively correlated with the percent crystallinity of the implant. So based on our research findings, we have several recommendations, we have the current thinking for the PhD development. We have several recommendations. Our first recommendation is use of type 1 collagen from the same species as RLD for the generic development. Additionally, a comparative characterization method should be established to demonstrate a sameness of the collagen to that of RLD. For example, hydroxypyrin content, glycine content, nitrogen assay, nitrogen over hydroxypyrin ratio, soluble collagen content. All of these parameters can be used to demonstrate the sameness of the collagen compared to the RLD. Of course, comparative physiochemical characterization of the test and RLD, including comparative in vitro release study, should be provided to provide the equivalence product performances between test and RLD. So in conclusion, we have already, from our research, we have already demonstrated that collagen source critically impacts the in vitro performance of the implants. Sameness of the excipients and in vitro BE challenges for demonstrating the product equivalency is feasible, and through establishing a comprehensive characterization framework. And whatever research I have provided today, shared today, and the part that I couldn't share due to lack of time, all the findings will be published in the peer-reviewed journals to facilitate science-based generic product development. And the data that we have generated through our research is continuously supporting the development of the PhD to support the in vitro BE assessment of the collagen implant. And then my final slide is acknowledging the wonderful team that we have, and the work was possible because of their wonderful collaboration. I want to acknowledge first my ORISE fellows, Shamika, Priyanka, and Ali, for their research support, and the rest of the team from OPQR, our ORISE colleague from OGD, for their continuous collaboration, ensuring that our research is aligned with the PhD development challenges, as well as helping us in drafting of the PhD. We are collaborating with them. And then our external collaborator, DGM, for their AI image analysis and segmentation expertise. And of course, the SME team involved in the STT meeting from multiple offices of the FDA, OPQR, OGD, OLDP, ONDP, OPPQ. Finally, the STT meeting chair for their great support and for taking this great initiative. With that, I conclude my presentation, and I will take the question during the panel discussion. Thank you. And next, I'm moving to Han.
Hello, everyone. I'm Liangfeng Han. I'm the clinical analyst at the team of nasal and orally inhaled drug product at the Division of Therapeutic Performance 1 Office of Research and Standards. Today, I would like to discuss FDA's option-based bioequivalence approach we recommended in the PSGs for locally acting metered dose inhalers and the dry powder inhalers. Years ago, conducting a clinical endpoint BE study was necessary for BE establishment, but we all know that the clinical study is time costing, the results is insensitive, and it's very expensive. Therefore, the agency recommends the alternative BE approach in lieu of the clinical study. In today's presentation, I would like to explore the available tools, supportive FDA research, and the external input for developing alternative BE approach, and the overview of the option-based BE approach for OIDPS, And our study design consideration for formulation sameness and determination, as well as several alternative PE studies, including the realistic APSD dissolution, particle morphology of the emitted dose, charcoal block pharmacokinetic B studies, and the computational modeling. I will also provide discuss the challenges and the future directions for OIDPB approaches. At the end of the talk, I will present the conclusions of our current thinking. So the alternative B studies ideally can probe each step of the local drug delivery, from the pre-actuation to post-actuation, from traveling through the airways to local lung deposition. Now we have a bunch of in vitro studies, including the realistic APSD dissolution and all listed here, and several in vivo study and the in silico studies. And all these studies that put together can investigate each step of the drug delivery process. And we think that the alternative B approach can provide a comprehensive evaluation of local drug deposition and for the B establishment. Therefore, OIS used the GDUFA funding to support a variety of research projects, trying to identify the factors that are critical for drug product performance. And these studies has provided us valuable information. For example, which alternative P study is critical for specific product, and the specific study design, and the pro and the cons of each study. Before 2008, almost all our PSGs recommended the clinical endpoint B study as a part of the B approach. Later, the agency started to recommend alternative approaches to the clinical study, either by an option-based B approach in option 1 in the PSG without the recommendation of clinical study, or the PSG does not recommend the clinical B study. In 2019, the first alternative B approach is that in the PSG for generics referring Cuva was available. In 2025, and there is 11% PSGs without the recommendation of clinical studies. And there are 58% of the PSGs with alternative approach available. So in total, we now have about 69% of the PSGs in which the clinical endpoint study is not necessary for B establishment. And on the right figure, you can see that in the recent 2 years, we have seen a dramatic the PSGs is dramatic increase of the PSGs in which we do not recommend the clinical. We do not think the clinical endpoint study is absolutely necessary for B establishment. So based on our research results and the feedback we received during the public workshops, we developed the option-based B approach. Here is the summary of the option-based B approach. For option 1, that waives the clinical endpoint study, we recommend that for the formulation sameness, no difference in formulation between the test and the RD products. In addition to the conventional in vitro studies and the conventional in vivo PKB studies, we also recommend alternative B studies, including realistic APSD dissolution and the PKB study with charcoal block. And if the product has a very complex formulation, we also recommend that the applicant to conduct a comparative characterization study, such as particle morphology of the emitted dose. And the firm also has the option to submit the computational modeling studies to support their BE approach. I want to note here that the choice of individual BE studies is really product specific. And if you have any questions, you can submit the meeting request to discuss your overall BE approach. And for option 2, that follows the traditional BE approach, including the clinical endpoint, we do not recommend that the formulation sameness. And similarly, we also, in some cases, you can submit the comparative characterization studies and the optional computational modeling studies. So for option 1, that our previous definition of formulation sameness is the test formulation is Q1 and Q2 the same as to the IS. And later we expanded the definition of formulation sameness a bit. So the test formulation either can demonstrate the Q1, Q2 the sameness to the IS, or if the formulation sameness not met, the firm can demonstrate the formulation changes will not affect the local and the systemic bioavailability of the API. The firm need to provide the justification, which may include but not limited to the formulation characterization data, product development data, comparative characterization studies, and the scientific literature. The goal of these additional studies is to understand the formulation design space of critical excipients and their ranges and their potential impacts on API bioavailability within the lung. The information data and all studies warranted will depend on the formulation changes being proposed. Ultimately, the firm should ensure formulation difference do not impact the desired outcome in product performance, safety, and efficacy. Now I move to the specific study design of each alternative BE study. Compared to the conventional APSD study, realistic APSD study incorporates more clinically relevant conditions via considering patient factors. The realistic APSD study used the patient breathing and the inhalation profiles rather than the constant flow rate, and used the representative mouth-throat models instead of the USP. We think that the realistic APSD can help us understand the impact of patient variability on aerosol performance. And the GDUFA-funded research has identified several factors that can affect the realistic APSD study. For example, the realistic mouth-throat models, inhalation profiles, and the model coating type, and the model insertion angle, and the MDI firing points. And we know that the response to these various study factors is product specific. In the figure on the right, you can see that the fine particle fraction variability for different the MDIs when different the mouth-throat model type and size are used. So we think that for your method development, you may consider these factors and for your studying. The PSG PSG recommended the realistic APSD conducted at the beginning stage, including the different mouth slot size and the inhalation profiles that reasonably covered the expected intersubject variability of the indicated patient population via bracketing approach. For example, small and large mouth slot model size and the weak and strong inhalation profiles that cover the patient population. And if you have the data to show the good correlation in vitro performance to the in vivo lung deposition data, you can provide this data to us and use the relevant the mouth slot model and the inhalation profile combination for the realistic APSD study. And we also recommend that the inhalation profiles should be obtained from patients, not from the health subjects. And the B of the realistic APSD is based on the population bioequivalence of impact size mass for each mouth slot model inhalation combination. And we know this recommendation involves a lot of statistical analysis. So if you have alternative statistical approach, so you can provide your justification and request a meeting to discuss your justification and your approach. The solution study helps to understand the rate at which the API dissolves. And the dissolution study is not recommended for every product. It's only recommended for the following two cases. For example, the API is dissolution limited and or contains other formulation properties that make dissolution the rate limiting step. The example list here, you can see that most of these APIs is not easy to dissolve. And we recommended the dissolution study for these APIs. The DUFA-funded research has helped us identify many contributing factors that can affect dissolution performance and the study sensitivity. We understand that currently there is no standard method, and the method development is product specific. And we expect that you can develop the dissolution methods that are sensitive and discriminatory to meaningful difference in the formulation and the manufacture process. The need for the dissolution study is API and the product specific. And the PSG recommended the dissolution study conducted at the beginning stage and collect the aerosolized dose of similar drug mass between the T and the IS product. And your method should be optimized and validated. For example, you have considered the optimized apparatus, sample collection, dose, media type and volume, agitation rate, and the sampling time. We think the critical that for the successful dissolution study is that the dissolution study should have discriminatory capacity. That is, your method can identify the difference in deposited drug particle size. The B is based on the comparative analysis of the dissolution profile, and we usually use the F2 analysis for the statistical method. Of course, if you have other methods, you can discuss with us to see if that's valid. For products with very complex formulation, for example, that the Topi halers, that this dry powder API has that porous structure. For this complex formulation, we recommend the particle morphology studies because the particle shape, surface roughness, porosity, crystalline and amorphous structure, etc. of the residual aerosolized dose once deposited in the lungs can impact the rate of dissolution of the API and the cellular permeability and uptake within the lungs. The firm should compare the residual particle morphology, agglomeration behavior, amorphous crystalline content, and polymorphs, and for those cases with complex formulation considerations. And we think that the advanced imaging method of the emitted dose can be used for the particle morphology study. And now we think that the comparative characterization studies provide supportive evidence for establishing BE between the T and the RS OIDPS. For example, the particle morphology can contribute to the APSD and the dissolution performance for certain OIDPS. Whether a PSG4 in OIDP incorporates comparative characterization studies depends on the specific product. And the figure on the left showing the results of particle morphology study, it indicates that the four different products with different amount of free fluticasylpropionate or the API agglomerate with the lactose. And these different level of agglomerations that can be detected by the dissolution study shown on the right, and with the highest that the agglomeration show the slowest that the dissolution rate. For the particle morphology study, the PSG recommends a minimum of 3 batches of the T and the IS product should be tested using the beginning life stage of the product. Imaging comparison should be conducted on the deposited particles of the emitted dose rather than the particle within the canister before the actuation. And the morphology features of the particles, which may include their agglomeration characteristics, should be evaluated. A description of the sample collecting method should be provided. OIDPS can be swallowed after oral inhalation. And if the swallowed portion showed significant GI absorption, the conventional PKB study without charcoal block cannot distinguish the systemic exposure contributed by inhaled portion or swallowed portion. For those products, therefore, we recommend that the PKB studies with charcoal block, which eliminates the GI absorption and it can provide the PK data from the inhaled portion. The PSG recommendation for the charcoal block PKB study is very similar to a PKB study without the charcoal block. For example, health adult male and female subjects can be recruited in the study, and the minimum number of inhalation to sufficiently characterize the PK profile with sensitive analytical method. Those administration should follow the approval labeling instructions. Bio-IND may be needed if the dose is above the maximum labeled single dose. And we know that there is no standard for charcoal dose, and the selected dose and how it is administered should be justified in the end submission. And the B is based on the 90% of confidence interval for the TR ratios of AUC and Cmax being between 80 to 125%. And the prospective applicants are encouraged to discuss other approach for assessing BE in local and systemic bioavailability of the API with the FDA via the pre-ENDA meeting request before conducting the charcoal block PKB study. For example, you can provide an alternative BE approach, the PKB approach, for example, the partial AUC, and to see if it fits for the BE established for your product. With respect to the computational modeling, we think computational models can provide support for a wide array of questions impacting both drug development and the assessment of performance. Now we have various in silico models, for example, regional deposition models and the CFD models and PBPK models available and can serve different purposes. Our PSG recommends that the applicant provide the purpose of your overall modeling strategy. For example, the impact of the product factors on regional drug delivery to establish biorelevance BE limits for BE studies and assess regional lung deposition BE via virtual simulations. And you also included that the model purpose should be well stated. For example, the CFD or semi-empirical model to predict the central and the peripheral lung depositions. PBPK models are useful if drug absorption is not expected to be rapid, such that the regional deposition may not be considered as a surrogate for regional lung delivery. And you also need to show that the model credibility and the validation established. Model verification is need to be established credibility. Model validation acceptance criteria and the statistical analysis method for virtual BE study should be defined prior to testing and should be justified. And although we have made a significant progress of the option-based BE approach, we think that we still have a lot of room to improve. For example, there's need for the method standardization for realistic APSD. We want to know which mouse slaughter models and the inhalation profiles to use for bracketing, and which combinations that reflect the in vivo situations that more accurately. For dissolution studies, we need to use standardize the sample collection, dissolution apparatus, dissolution media, etc. For charcoal PKB studies, and we need to standardize the charcoal dosing for most products. We also need to establish validated in silico models to support BE evaluation. And we also need to find the area to streamline and harmonize BE approach globally for OIDPS, for example, to identify key and supportive BE studies and to establish the in vitro and the in vivo relationships. We think our improvement in these areas can help us improve the BE establishment and reduce unnecessary testing. And FDA will continue its effort to address these challenges, and we recommend you refer to the most recent PSGs on MDIs and DPIs, which are updated periodically and will reflect the agency's current scientific thinking, and the prospective applicants are highly encouraged to discuss their development plan with the agency to gain feedback on complex and challenging issues, including the appropriate study designs, analysis and the supporting justification for new next generation per-patent MDI submission. In conclusion, to address these challenges, FDA has explored in vitro, in vivo, and in silico studies designs through GDUFA-funded research initiatives and workshops to identify alternative approaches that can be used in lieu of the clinical endpoint B study for establishing local drug delivery equivalence. FDA recently developed PSGs for MDIs and DPIs that recommend an option-based B approach. Study recommendations are included for realistic APSD dissolution comparative characterization studies, charcoal block PKB studies, and the computational models. While FDA's option-based B approach allows for B pathways without a clinical endpoint B study, FDA will continue its effort to clarify, streamline, and harmonize its B approach for OIDPS. So I would like to acknowledge our FDA colleagues across all different office and divisions and our external research collaborators, and put together their work that allow us to stand where we are at now. And thank you very much. And I will wrap up my talk now here and turn to the next speaker, Megan. Thank you.
Thank you, Dr. Han. Hello, my name is Megan Kelchin, and today I'll be discussing how our office is streamlining recommendations for topical products. And for the purposes of today's presentation, I'll be focused on topical products that are applied to the skin or the mucosa, such as vaginal products or rectal products. So I'll start by discussing an overview of bioequivalence approaches for these topical products, and then dive a little deeper into the waiver approach for BE studies for additional strengths for the topical semisolids, and then finish with some practical insights on ANDA submissions. So based on GDUFA-funded research, OGD has developed efficient characterization-based bioequivalence approaches for topical products applied to the mucosa or the skin. And we've incorporated these efficient characterization-based approaches into our product-specific guidances. I've already done a deep dive into the components of the characterization-based approach in previous presentations, so if you'd like to learn more, I encourage you to review those presentations. But for the purposes of today's presentation, I'll briefly note that in our PSGs for the characterization-based approach, we recommend no significant difference in formulation compared or between the test product and the RS, comparative Q3 characterization, and an in vitro release study. And depending on the complexity of the dosage form, we may also recommend an IVPT study or other biorelevance study or a PK study. In addition to the characterization-based approach, we often also recommend in vivo BE approaches in our product-specific guidances. And two of the common in vivo BE approaches for topical products is a comparative clinical endpoint BE study, which compares the efficacy of the test product and the reference product, and both are shown to be superior to a placebo control, or a vasoconstrictor study, which is a pharmacodynamic study that evaluates the skin blanching effect of topical corticosteroids. So as I mentioned, over the past several years, we've been incorporating efficient bioequivalence approaches into our product-specific guidances with the goal of incorporating an efficient BE approach in every PSG for topical products. Dr. Kumari has just discussed how research can be utilized to develop bioequivalence approaches for newly approved NDA products, but I also want to highlight that we can utilize research to support the development of efficient bioequivalence approaches for products where the PSG recommends a comparative clinical endpoint study. So as an example, I want to use roxalate phosphate topical cream. This is a drug product where we had internal research with our FDA labs. And as part of that internal research, we were able to develop a characterization-based bioequivalence approach for this drug product. And as a result, we revised the PSG in November 2024 to incorporate the characterization-based approach as an alternative to the comparative clinical endpoint bioequivalence study. Now, research is not just limited to the newly approved NDAs or recently approved NDAs. We also utilize research to revise the PSGs for older products. So as an example, terbinafine hydrochloride topical cream is an older product where the PSG had been previously revised in February of 2019 to include a comparative clinical endpoint study. And based on a decade of GDUFA-funded research, we were able to incorporate a characterization-based approach into the PSG in May of 2025. So as you can see, we're continuously adding efficient bioequivalence approaches to PSGs, and this is based on scientific innovation with our external and internal research collaborators, availability of resources, and interest from the industry. While we have limited resources available to develop and revise these PSGs, we rely on the industry to help drive which products we should prioritize for drafting and revision. And so if you have a drug product in mind, but the PSG recommends an inefficient bioequivalence approach, such as a comparative clinical endpoint study, we encourage you to come to speak with us as part of the pre-ANDA product development meeting process. And of course, you can always review our upcoming PSG website to get a list of the products that we plan to, PSGs that we plan to work on in the next 12 months. So now I want to turn to a specific enhanced bioequivalence strategy, which is a waiver of BE studies for an additional strength when the RS product has multiple strengths available. You'll see in the coming slides that I focus particularly on situations where there are 2 strengths of the RS available, but I want to highlight that this waiver approach may also be applicable for products where there are more than 2 strengths available. So historically, OGD has recommended resource-intensive in vitro or in vivo studies for each strength of the product to support a demonstration of bioequivalence. So using tacrolimus topical ointment as an example, the PSG that was published in October of 2022 recommended for the higher strength a characterization-based approach that included an IBPT study or a comparative clinical endpoint study. And if you look at the PSG for the lower strength, we recommend those exact same studies. And so if you intended to develop both strengths of tacrolimus topical ointment, you were expected to do all of the studies that were outlined within a given approach for each strength. So that means if you use the characterization-based approach, you would need to do all of the studies for both the higher strength and lower strength. And if you use the in vivo approach, you would need to do 2 comparative clinical endpoint studies. Based on internal research, we are now able to streamline our recommendations and waive a BE study for an additional strength, provided that the conditions of the waiver are met. And I'll discuss those waiver conditions in the coming slides. But as an example, you may be able to waive an IBPT study when using a characterization-based bioequivalence approach, or you may be able to waive a comparative clinical endpoint study if you use the in vivo approach. So going back to tacrolimus topical ointment, we revised the PSG in November 2024 to incorporate this waiver approach. And as part of the waiver approach for these products, when using the characterization-based approach, you may be able to waive the IVPT study for the lower strength. And if you use the in vivo bioequivalence option, you may be able to waive the comparative clinical endpoint study for the higher strength. So shown here is the general framework for the waiver approach for topical products. And this generally follows the same framework that we use for waiving bioequivalent studies for oral, solid oral dosage forms. So in general, the recommended strength of the test product is demonstrated to be bioequivalent to the recommended strength of the RS. And the recommended strength for a given BE approach is described in the product-specific guidance. Following demonstration of bioequivalence, the additional strengths of the test product are compared to the recommended strength of the test product. And in general, the relationship between the 2 strengths of the test product should be compared to the relationship of the different strengths of the RS product. So next, I'll go into the components of the waiver approach. The first component is demonstration of bioequivalence of the recommended strength. So when using a characterization-based approach, we recommend demonstrating bioequivalence to the RS using the high strength and then waiving the IBPT study for the lower strength. When using an in vivo approach, the strength that is recommended for demonstrating bioequivalence is product-specific, and so I refer you to either the PSG for that product or come in and talk to us as part of the pre-ANDA meeting process before initiating your in vivo studies. The next component is related to the formulation and manufacturing process. The formulations of all of the strengths of the test product should be exactly the same, except for the amount of drug and the corresponding amount of the diluent. Additionally, the same manufacturing process should be used for all strengths of the test product. Next is comparative Q3 characterization of each strength of the test product and the RS. And in general, the relationship of the Q3 attributes for the two strengths of the test product should be compared to the relationship of the Q3 attributes of the two strengths of the RS. Now, for some of these Q3 attributes, such as pH, we expect that there won't be much difference between the different strengths of the test product. However, for other Q3 attributes, such as globule size distribution, which may impact the microstructure of the drug product, we want to ensure that the relationship for the between the strengths of the drug products is maintained for the test product and the RS. Finally, we recommend IVRT studies for each strength of the test product and the RS. One way to evaluate this IVRT data is to compare the relationship of the release rates of the different strengths of the test product to the relationship of the release rates for the different strengths of the RS. Alternatively, if utilizing a characterization-based approach, the test product would meet the no significant difference standard in formulation compared to the RS. And in that situation, you may be able to compare the release rates of the test product and the reference product for the recommended strength and the test product and the RS for the additional strengths. So shown here is just a summary of the components that I just went over for the waiver approach. But what I want to emphasize with this slide is that in your ANDA submission, you should include comparative Q3 characterization data and IVRT data for each strength of the test product and the RS. So next, I'll briefly dive into a little bit deeper into the IVRT studies with the focus on IVRT method development and validation. In general, we can use the test product to validate the IVRT method, but I want to highlight that if your test product does not meet the no significant difference standard compared to the RS, we recommend that you ensure that your developed IVRT method is suitable for both the test product and the RS before you move into full-scale IVRT method validation studies. We recommend that the IVRT study or the IVRT method is validated for all strengths of the drug product. And if you are not developing all strengths at the same time, I want to note that you may need to repeat some of the method validation studies, especially if significant time has passed between the development of the different strengths. So now I'll move into some practical insights for ANDA submissions. And the intention of providing these insights is to provide tips that may help minimize delays in your product development in ANDA submission and minimize the need to repeat studies. So as I mentioned at the beginning of my talk, PSGs that include a characterization-based approach recommend no significant difference in formulation. And you're able to confirm that your proposed test formulation is eligible for the characterization-based approach in a controlled correspondence before conducting your Q3 or bioequivalence studies. It's important to note that all relevant information related to your proposed test formulation should be included in your controlled correspondence. And so I want to highlight and the inclusion of all of the relevant information may minimize the need for information requests, and in turn would reduce delays in responding to the controlled correspondence. So I want to highlight the draft guidance that's cited on the screen here that's related to content and format of composition statements. And I want to specifically highlight this table that's included in the appendix of that guidance, which may be a useful guide in structuring your formulation table before you submit your controlled correspondence. So shown here is a common response that we often give for formulation assessments And as part of that response, we state that the characterization-based approach may be appropriate to support a demonstration of bioequivalence for your proposed for a given test formulation. Another common approach another common response that we've been providing looks very similar to the response I just showed. However, it includes an additional sentence that states that we strongly encourage you to review the agency's inactive ingredient database. And so what this means is that within the scope of the control correspondence, we were unable to assess if the proposed concentrations for a given formulation are acceptable for an ANDA submission when taking into consideration the context of use of the drug product. So what does this mean? This means that we encourage you, before moving forward with your studies, to submit a new control correspondence to confirm that the levels of inactive ingredients are suitable for an ANDA submission prior to conducting those studies. And this is important to, and this is also important for formulations that are intended to be used for in vivo bioequivalence studies. We strongly encourage you to confirm the levels of inactive ingredients for your proposed formulation are acceptable for submission in a prospective ANDA. And we recommend doing this prior to conducting any of the in vivo bioequivalence studies, because what we don't want to happen is you conduct your expensive in vivo bioequivalence study, and then we realize after those studies are wrapped up that your formulation may have some issues and you may need to repeat that study. Additionally, we encourage you to maintain photographic evidence documenting the impact of treatment at baseline and at the end of treatment when possible. And an example of where this might be applicable is a comparative clinical endpoint bioequivalence study for acne products. So now I want to briefly touch on TDS products. As part of internal research that we've done within our office, we reviewed the TDS ANDAs that were received during GDUFA 1 and GDUFA 2 to see if there was any signal that certain inactive ingredients in those drug products were associated with failures in irritation and sensitization studies. And of the 30 TDS products that we reviewed, we did not find any indication that the inactive ingredients were associated with irritation or sensitization. And I want to highlight a sentence in the guidance for industry related to irritation and sensitization potential in ANDAs, which states that an in vivo sensitization evaluation of a TDS product may be unnecessary if adequate justification is provided. And so if you have a question on whether your proposed test or proposed formulation for a TDS product may not require a sensitization study, we encourage you to submit that proposed test formulation in a controlled correspondence or a pre-ANDA product development meeting request and discuss with us on whether the sensitization study is needed. Finally, we recognize that there are challenges with IVRT and IVPT studies, and I want to highlight the BE study protocol review as a venue to discuss these challenges related to these studies, particularly related to the IVRT and IVPT method development and method validation studies. In your submission, you should include the method development and method validation reports and any data that you might have available that illustrates the challenges that you've observed with these studies. So in summary, PSGs for topical products evolve over time to incorporate efficient bioequivalence approaches based on cutting-edge research. The waiver approach for topical products, when there are 2 or more strengths available, can significantly reduce the regulatory burden to support the approval of multiple strengths of complex, locally acting semi-solid drug products. An engagement with the agency to gain feedback on proposed formulations and bioequivalence studies prior to ANDA submission can be beneficial. With that, I'd like to acknowledge all of my colleagues at the FDA who have contributed to this work in our development for the PSGs. And here's a list of resources that you might find helpful. With that, I thank you for your attention, and I look forward to answering any questions that you might have during the panel session. Thank you.
Thank you, Megan, and thank you to all of our Session 1 presenters. Your insights on product-specific guidances for complex products have set the stage for a rich panel discussion after our morning break. We will now take a short morning break and we will reconvene at 11:15 a.m. Eastern Time for our Session 1 Q&A panel discussion. During the break, you may submit your questions through email at orsevents@fda.hhs.gov with the subject line AGDD 2025, and we'll try and address as many of your questions as possible. We will leave this slide up for you to be able to reference that, and we look forward to welcoming you back at 11:15 a.m.